Hello, everyone. Welcome to today's lecture. Today we're going to be focusing on a psych-soc topics. That'll be sensation, perception, and intention. My name is Megan Burnson, and I'm one of our MCAT tutors with Socratic Med, focusing on biochem, and psych-soc. So here I just have linked our GroupMe. This is where we'll post links to our lectures and where you can also ask any questions that you might have about lectures, and one of our tutors will be able to get back to you. Our Instagram is here as well. Our YouTube channel where all of our videos are uploaded from both this 15-week question-based learning course and all of our other courses as well. So there's a lot of great material on there to help you with any subjects that you might need to focus on. So our schedule for this question-based learning course is 2 lectures per week. So we have one lecture on Sunday evenings that is released at 8:30 p.m. Eastern, and then Monday evenings as well, also at 8:30 p.m. Eastern. So we could just jump right into it, and here we go. So question 1 is going to be focused on sensation. And the question reads, which of the following receptors consists of a free nerve ending that detects chemical signs of tissue damage? So take a minute here and pause the video and answer your question. All right, so assuming that you've answered the question now, I'll show you the answer, and that is going to be B, nociceptors. So we're going to go through and talk about all of the different types of receptors, including chemoreceptors, thermoreceptors, mechanoreceptors, and nociceptors. And these will be our pain receptors. All right, so just a quick introduction to sensation. So sensation refers to the ability to detect or sense the physical qualities of our environment. So typically you'll think of like the 5 senses, so sight, hearing, taste, touch, and smell. So we have exteroreceptors, which detect stimuli from the outside world. So things like sight, hearing, taste, those are all outside stimuli. And then interoreceptors will detect stimuli from internally. So things like pain that you feel within your body, when you feel like your stomach grumbling or your stomach hurts or your head hurts, those are all internal stimuli that are causing those sensations. So the different types of receptors that are involved in sensation are mechanoreceptors, chemoreceptors, nociceptors, thermoreceptors, and electromagnetic receptors. So we're just going to go through these 5 types of receptors really quickly, just talk about how they all work and some examples of where they can be found. So mechanoreceptors, a lot of these are present like in the skin or near the skin surface, and these respond to changes in pressure or mechanical disturbances. So we have Merkel's discs right here in this image, and those respond to light touch. We have Ruffini endings, which respond to stretching. Meissner's corpuscles respond to touch and low frequency vibrations. Pacinian corpuscles respond to transient pressure and high frequency vibration. And then auditory hair cells and vestibular hair cells are also mechanoreceptors, and those are involved in audition or hearing and balance. So the auditory hair cells will be present in the cochlea, and the mechanical bending of those hair cells will cause them to fire, and vestibular hair cells are in our semicircular canals, and movement of those cells will contribute to our ability to balance. So chemoreceptors are receptors that will respond to different chemicals. So an example of these are olfactory receptors. So that would be in the nasal cavity, and they respond to airborne chemicals that allow us to sense different smells. And then gustatory receptors will respond to different chemicals within the mouth and allow us to have a sensation of taste. So our taste buds are gustatory receptors. So these are just some examples of chemoreceptors, pretty straightforward. So nociceptors, these are our pain receptors, and they're the simplest type of sensory receptor because it's essentially just a free nerve ending that responds to signs of tissue damage. So that's how you know that there should be pain there because something's wrong, something's damaged. That's why these receptors transmit pain. So this can be either somatic or autonomic response, and it can kind of be thought of as a simple chemoreceptor because what it's detecting is technically chemicals, like chemical changes involved with tissue damage. So thermoreceptors are receptors that respond to changes in temperature. So we have cold sensitive receptors that will respond to colder temperatures. Our warm sensitive receptors will respond to higher temperatures. And our thermal nociceptors, that is when you detect painfully hot stimuli. So when you burn your hand, it's a thermal receptor, but it's also a nociceptor. This can also be autonomic or somatic. So electromagnetic receptors respond to electromagnetic waves. So there's only 2 examples of these in humans, and that's going to be our rod and cone cells of the retina. So that will be responsible for vision, and those can also be called photoreceptors. In some other species, you can have separate electroreceptors and magnetoreceptors, but in humans, they are just electromagnetic, and that's our rods and cones. So an example of a magnetoreceptor in a different species can be the ability for something to detect the Earth's magnetic field during migration. Obviously, that's not a receptor that humans have, but there are some species that are able to detect that. All right, so question number 2, we have sensation that we're going to talk about. All right, which of the following components of sensory stimuli is encoded by the frequency of the action potentials? So take a minute here and pause the video and answer our question. All right, assuming you've answered the question, I will show you the answer now, which is going to be D, intensity. All right, so stimulus modality, that is something that is coded by the type of receptor responding to the stimuli. So that's just knowing, like, what are we sensing? Are we sensing, are we seeing something? Are we hearing something? Are we sensing like electromagnetic waves like in vision? Or are we sensing like something like touch? So that just depends on all those different types of receptors that we just talked about, whichever one is responding, like nociceptors. Those will tell us that the stimulus is pain and things like that. So stimulus intensity is encoded by the frequency of action potentials. So basically just if you keep firing, that means it's a more intense stimulus than if the frequency was lower. So stimulus location depends on where that sensory receptor is in the body, and that can also be improved by neighboring receptors being in the same location that will also fire action potentials. So that can help us to localize things, especially like touch, to know exactly like what part of the skin is being touched. It depends on not just the exact place, but surrounding receptors can also be firing. And this can be made more specific by inhibiting surrounding receptors. So if it's like really precise, then sometimes the direct receptors around wherever the stimulus is coming from can be inhibited. So that just makes it a little bit more specific as to where it's coming from. So stimulus duration, this isn't always encoded explicitly. So tonic receptors are receptors that fire action potentials for the duration of the stimulus. So these will actually encode your stimulus duration, where phasic receptors only will fire an action potential at the beginning of a stimulus and will not communicate the duration. So that's only if the stimulus is a constant intensity. If it's not changing, then these phasic receptors won't keep firing. But if stimulus gets either more or less intense, then these phasic receptors will adapt. Yeah. All right, so on the topic of adaptation, a decrease in firing frequency for a stimulus with constant intensity is what is known as adaptation. So basically, you could think about just tuning out ongoing or unimportant information. So you're not always aware of everything that's going around. You probably don't sit there and think about the fact that you can feel your sweatshirt against your skin. Like that is not necessarily important information. It's not portraying anything that we need to attend to. So adaptation kind of explains this by basically saying that these receptors stop firing to a constant stimulus because it's essentially just background information and allows us to devote more attention to the more important things. So yeah, an example I have here is like when you walk into someone's home and notice a smell, say they're baking cookies or something, you'll definitely notice it right when you walk in. But when you're there for a few minutes or an hour, you probably won't notice the smell of the cookies anymore because it's not really changing in intensity. So those receptors will stop firing to tell you that there's a smell. And that's not to say that the smell went away or anything. If someone else came into the house, they would still say, wow, it smells like cookies. And you're like, oh, I don't smell it anymore. So just a good example to sort of give you some context and to be able to think about this in your real life to help you remember it a little bit. All right, so question 3 is also going to be about sensation. All right, which of the following is involved in proprioception? So our numbers here are muscle spindles, Golgi tendon organs, or joint capsule receptors. So take a minute here and pause the video and try to answer this question. All right, so assuming you've answered the question now, I will show you the answer for this one, and that's going to be D, 1, 2, and 3. So all of these are involved in proprioception, which we'll talk a little bit more about now. So proprioception is the awareness of the body position. It can also be called kinesthetic awareness. So basically, like, it's not something that you really think of when you think of senses, but like if you're holding your arm out over here, your brain is telling you, like, you know, your hand's out there and you're aware of where that body part is, just as you are with the rest of your body. So this sensation relies on the following receptors. So that's muscle spindles, which are mechanoreceptors that detect muscle stretch. Golgi tendon organs monitor tension within the tendons, and joint capsule receptors detect movement, tension, and pressure in the joints. So these are just a couple receptors that are involved in the sensation of proprioception. It's definitely good to be familiar with them, how they differ from each other, and kind of their role. All right, so now we're going to go through and just go over the other, the 5 senses. So we're going to start with gustation or taste. So taste receptors are able to distinguish between the following 5 flavors, and that's sweet, salty, bitter, sour, and umami. So here on the right, I have a picture of a taste bud structure. So the taste bud is composed of epithelial cells. The taste pore is this part down here, which is just the big center part of the taste bud. And then the taste hairs up top are what detect the food chemicals. So the sensation of taste is transmitted to the brain by cranial nerves. So olfaction, this is our sense of smell. So we have olfactory receptors that are present within the nasal cavity, also known as the nasopharynx. So these again are going to be an example of a chemoreceptor, just like gustation was. So olfactory nerves project to the olfactory bulb within the temporal lobe. And olfaction is also very involved in our interpretations of taste. So if you think about like when you have a really bad cold or something, you're really congested, and you can't really smell, you also tend to lose your sense of taste. So now with COVID too, when people are losing their smell, they're also entirely losing their taste, which definitely has some importance and is something that you should definitely be aware of. All right, so I'm going to talk about audition or hearing just pretty briefly now. This will be covered more in depth in week 13. So I think that's 5 weeks from now. But yeah, so the outer ear is composed of the pinna, and the auditory canal or the ear canal. So that is all external, and those are the two structures that make up the outer ear. So the middle ear is divided from the outer ear by the tympanic membrane. So that's sort of our split between the outer and middle ear. And then the malleus, incus, and stapes, those are the three smallest bones in the body, and they transmit the pressure waves from the eardrum onto the oval window. The oval window is on the cochlea, and that is our division from the middle ear to the inner ear. So we have our cochlea, which is this snail-shaped tube, basically, that has those auditory hair cells that we talked a little bit about earlier. And there's like a thick jelly-like fluid, and the sound waves that are transmitted by the stapes move that fluid, and that's how we're able to detect different sounds or different frequencies, things like that. The semicircular canals up here, those have those vestibular hair cells that help us with balance and movement. And then the utricle and saccule are also involved in balance and movement. So again, we'll talk about all of this a lot more in depth in week 13, but I just wanted to cover it now while we're talking about the other senses. Same with vision. This will also be covered in week 13, but I did want to just touch on it a little bit now. So yeah, here are just some structures of the eye. So we have our cornea, which is the outermost structure, and behind the cornea is the aqueous humors. So this is a liquid that is present there. Sorry if you hear my cat meow. And then we have the iris. So the iris is like the colored part of the eye. So muscles in the iris determine the size of the pupil, and the pupil is where light will actually pass through. So for as you can see here, we have this object, and then the pupil size is accommodated so that the light can pass through and focus that image on the retina. So we also have our lens, which is something that is controlled by these ciliary muscles, and that helps us focus on like near or far objects. So it basically bends, and that elasticity of the lens is something that you actually lose with age, which is why many older adults will need reading glasses, because their lens is not able to accommodate as easily as a younger individual. So on the retina, we have those rods and cone cells that we talked about earlier, and those are electromagnetic receptors. So those will respond to light. And then we have the optic disc, which is in the back of the eye where the optic nerve leaves and goes to the brain. And the optic disc is actually where your blind spot is. So if you ever like have done the blind spot tests, you can probably just Google it. I know some textbooks talk about it a little bit in like maybe an intro psych or bio class. But since, yeah, there's no photoreceptors there because there needs to be a place for that optic nerve to leave. So that is why we have a blind spot. All right, so question 4, we're going to talk a little bit about perception. So which of the following is an example of a binocular cue? So take a minute here and pause the video and answer this question. Alrighty, assuming you've answered the question, I'll show us the answer, which is B, retinal disparity. So interposition, relative clarity, and texture rating are all monocular cues. So they only rely on one eye. So we're talking about cues of basically like depth perception in vision. So binocular cues will use both eyes, and monocular cues will only rely on one. So the two binocular cues that we're going to talk about are retinal disparity and convergence. So retinal disparity is basically the ability of the brain to compare the images on the two different retinas. So obviously this involves both eyes because it's comparing the two images. So a good way to sort of understand this concept is by doing this test where you basically hold your thumb out in front and you cover one eye and look at it, and then you cover the other eye and look at it. And you'll see depending on how close it is, how much that thumb will move between those two eyes. So if it moves more, that means that the object's closer. If it moves less, it means the object is further. So that's one way that our brain is able to tell how far away an object is. You're not really aware of the fact that this is happening unless you do that thumb thing that I just talked about, but it is. So yeah, that's just one way that we can tell how far away something is. So convergence is kind of similar, but it's just the extent to which the eyes turn inward. So the more that they're turning inward, the closer an object is. You can also test this using your thumb. So if you just keep bringing it closer to your face, you'll definitely notice that your eyes at some point start straining to focus inward and actually focus on that thumb when it gets closer. So yeah. Monocular cues. So these are cues that only depend on one eye, and I have some pictures over here that kind of demonstrate them. So interposition, in this image it says occlusion, means the same thing, but that's when a nearer object blocks the view of a farther object. So here, yeah, it is just showing that like this blue circle would be perceived as closer than the green circle because it's covering it. So relative clarity, it's not pictured over here, but you could just know that if something is closer, you'll be able to see it more clearly. Something is further and a little bit blurrier, then that will be seen as further away. Same with relative size. So if two objects are known to be the same size and one is smaller than the other, that will be perceived as further away. Relative height. So usually when something is lower in the visual field, it's closer. Something's higher in the visual field, it's further. So to remember this, I usually think, I kind of coupled this with relative height, or not relative height, linear perspective. When I think about like doing art in elementary school and you had to draw a landscape, like everyone would draw a road going off and it would always have that kind of like typical shape where it turns into a little triangle and disappears. So that shows like linear perspective, and it also shows that the higher in the visual field, the further away, because that always goes up towards usually the middle of the page, then you have your sky or whatever. Yeah, so relative motion, this isn't really, this is more in like real life, not so much something that you can see in an image, so it's not pictured on the right here, but stable objects that are moving faster with your movement are closer to you, whereas really distant objects probably won't really look like they're moving at all if you're just walking or something like that. So yeah, it's another monocular cue. Texture gradient, that is pictured here. So more distinct texture means something's closer, and indistinct texture increases with distance. So it's kind of similar to relative clarity. The further something away is, the less details you'll be able to see. So light and shadow is another way we can see the position of objects. Closer objects will appear lighter in color. And then linear perspective, that's what I was just talking about, how lines that are parallel will appear to converge or come together with increasing distance. All right, so question 5, we're going to talk about perception a little bit more. So which of the following terms is most related to the idea that we use prior experiences to impose expectations onto a certain stimuli? So take a minute here and pause the video. All right, so our answer here will be C, top-down processing. So we're going to talk a little bit about the Gestalt laws of grouping and signal detection theory. Those aren't really involved with the question, so you really should have been between bottom-up and top-down processing. So bottom-up basically is saying that we just detect the raw stimuli and that gets transmitted and then we interpret it. Top-down basically is saying that we use prior experiences and impose our expectations onto a stimuli, and that helps us perceive it a certain way. And in reality, we actually use a little bit of both of these, so it's not black and white, one or the other. It kind of depends on the situation too. So yeah, we'll just talk about these topics a little bit more in depth now. Yeah, so top-down processing, this is applying previous experiences, knowledge, or expectations in order to interpret sensory input. So a good example of this is the Stroop effect, which you may or may not have seen before, but it's where you're directed to say the color of the word, not the word, or vice versa. So this shows that it's not strictly going to be bottom-up processing, because a lot of people have difficulty with this task, especially trying to do it quickly, because you either get distracted by the word itself or by the color of the word. So if you were just reading the word, it wouldn't matter if that the word purple is the color red. But since you have the expectation already, you're looking at it, you know it's red, you want the word to say red, and it doesn't. So yeah, the bottom-up processing is pretty much the opposite, and just says that we begin with our sensory receptors and work upwards from there to integrate that information. So this approach is used when we have little to no experience with a stimulus. So if we're unable to impose our expectations or anything onto that stimulus, then we will be using bottom-up processing. So yeah, what I said earlier is important to note, that we use a combination, both bottom-up and top-down processing to interpret and perceive different stimuli. So Gestalt laws of grouping. This will make a little bit more sense when I show an image on the next page, but I'm just going to go through these now. So the law of proximity states that things that are near each other tend to be thought of as grouped together. The law of continuity is the idea that we perceive continuous lines and forms rather than broken ones. So if we're able to see something as a continuous line, that's usually how our brains will try to perceive it. The law of closure is the idea that our brains will fill in gaps to create something that's complete. And the law of connectedness is the idea that when things are linked, they are perceived to be connected. Yeah, so we have some picture of that here. So proximity, so like these two lines and these two lines all seem to be grouped together. So you would think of these as like groups of two. Similarity, you would group these triangles, group the circles, and group those triangles. Continuity, you perceive this as like a straight line with a curvy line going through it, instead of like a bunch of little semicircles and a bunch of single lines. Oops. Closure would be the idea that if we perceived all of these as a closed square rather than these bracket shapes, and then connectedness would be the idea that we see all these dots as connected, not just as a dot and a line and a dot. So yeah, hopefully that makes a little bit of sense now. So signal detection theory is an attempt to predict how and when a stimulus will be detected amongst noise or other sensory stimuli. So yeah, if you are testing this experimentally, there are 4 possible responses. So basically, if the signal is present and it is detected, that's considered a hit. If the signal is absent and someone thinks it's detected, that's a false alarm. If the signal is present and it is not detected, that's a miss. And if a signal is absent and not detected, that would be a correct rejection. All right, so another question about perception now. We have a fun little optical illusion in the back. All right, so Weber's law dictates that 2 stimuli must differ by a constant blank in order for their difference to be perceived. So take a minute here and pause the video to answer this question. All right, so I will show you guys the answer to this one now. The answer is B, proportion. So yeah, pretty straight up. Weber's law dictates that 2 stimuli must differ by a constant proportion in order for their difference to be perceived. So difference threshold can also be called the just noticeable difference, or JND. So this is defined as the minimum level of stimulation required for a noticeable difference to be detected 50% of the time. So as we just said, Weber's law states that two stimuli must differ by constant proportion for there to be a perceivable difference. And there are different proportions for different properties. So for weight, for humans, things need to differ by 2% for them to be detected as different. Light needs to differ by 8%, and sound needs to differ by 0.3%. So an example for weight would be if you have a 100-pound weight, a 101-pound weight, and a 102-pound weight, you would be able to tell the difference only between the 100-pound and the 102-pound weight, because those differ by exactly 2%, but the 101-pound weight would feel the same way as both the 100 and 102-pound weight. So I just thought that is a good example, a good way to think of that. So the absolute threshold is defined as the minimum input required to activate a receptor half of the time. So this is just to get any activation at all. So this can vary both between individuals within a species, and is often different between species. So an example of this would be like dogs are known for having a great sense of smell. So they have a much lower absolute threshold for smell than we do. So this is also something that can change with age within an individual. So as you get older, an example of this is losing your ability to detect higher pitched sounds. So the absolute threshold for sound, for the frequency will actually decrease, so you won't be able to detect those really, really high frequency sounds. And that doesn't have to do with volume or anything, it's the pitch. So the high frequencies will not be detected in older adults. Oops. All right, question 7. We're going to talk a little bit about attention now. So which of the following theories, assuming auditory processing occurs after filtering and therefore does not account for the cocktail party effect? So sorry, there's a typo in this question. It should say which of the following theory assumes auditory processing occurs after filtering and therefore does not account for the cocktail party effect. I apologize for that, but question is still the same. So we'll just take a minute here and pause the lecture and answer this question. All right, so our answer here is A, the Broadbent filter model of selective attention. So I think that was like one of the first models that came out that attempted to explain how things get converted into short-term or working memory in terms of attention. Treisman's attenuation model basically is very similar to Broadbent's, but it does attempt to account for the cocktail party effect. So the spotlight model is just not involved at all in attention, and it's not something that we're going to talk about. And then selective priming is involved with attention, but it also is something that's used to account for the cocktail party effect. So we will talk a little bit about that now. All right, so selective attention is the ability to attend to one input and tune out the rest. So the dichotic listening task is a way that we have tested this experimentally before. So it's a means of studying attention capacity where the participant here wears headphones and each ear has a different input. So they're directed to attend to one input, not the other. So that will be the attended channel is where they're told to listen to the message, and the unattended channels where they're told not to listen. So this test usually shows that people are unable to recall anything from the unattended ear, and shows that we definitely do have selective attention, and we have to focus our efforts on whichever the attended channel is, and we can't take in all sensory information at once, and we can't put it all into working memory. So the Broadbent filter model of selective attention was created by Donald Broadbent, and he created this in an attempt to better understand how memories are formed from raw sensory information, or how we are able to filter which information is remembered and which is not, like in the dichotic listening task. So he postulates that information first goes through a sensory buffer. This is where it is filtered and then sent to short-term memory. So basically in his model, it's saying that anything that is not attended to is filtered out completely in the filter and is not it's not sent to short-term memory at all. So this does not account for the cocktail party effect because so the cocktail party effect, let me just explain this first, is basically your ability to hear something notable such as your own name throughout background noise that was previously unattended. So it's called this because if you're at a cocktail party, you're having a conversation with someone and you're not listening to other people's conversations, you're just doing your own thing, but you hear your name somewhere off in the distance, usually that will cause you to be like, wait, I heard my name. So this model does not take that into account because it assumes that that information is all being filtered out and cannot be attended to. So yeah. So here's just a little image that kind of shows what this model is. So the selective filter basically completely blocks out any unattended information. So it bottlenecks all of the sensory input that you're getting and only sends the attended message to higher level processing, which is then sent to working memory. So Treisman's attenuation model is sort of an adaptation of this, but it attempts to take into account the cocktail party effect. So the main difference here is that rather than a selective filter, it has an attenuating filter, which sort of like turns down the unintended information rather than completely silencing it. So that is why with this one, you would be able to pull your name and that's out of like background noise, you'd be able to hear that even though you're not attending to that information because it's just turned down, it's not completely filtered out. So again, an image here where we have the attenuating filter. So it does still kind of have a bottleneck, but there is, yeah, there's like different thresholds here to what will get processed or not. So that's where something like your name, something really familiar and relevant to you, could be pulled out of the unattended message. And that can still be sent to working memory. Yeah, so here I just have these two compared so you can see them side by side. And yeah, the main difference again is that attenuating filter or the selective filter. So it's pretty explanatory by the name. Because we have the attenuation model has the attenuating filter, and then the filter model has the definitive or the selective filter. So selective priming is kind of relevant to Treisman's model. So it can also be used in explaining the cocktail party effect. So this is defined as the idea that previously encountered stimuli or expectations can make someone more likely to notice it when it does occur. So something like your name is definitely previously encountered. It's definitely very familiar, and something you're always kind of listening for. Like if that's how people get your attention is by calling your name. So you're kind of selectively primed to attend to your name when it is said. So yeah, this is just good to be familiar with the idea of priming. All right, so question 8 now, we're going to talk a little bit about attention. So which of the following is not associated with performance on multitasking? All right, so pause the video here and answer the question. All right, so I'll show the answer now, which is going to be D, none of the above. So all of these three, task practice, task similarity, and task difficulty, those are the three factors that play into your performance or your ability to multitask. So yeah, multitasking obviously concerns our ability to perform multiple tasks at the same time. So on the right here, I have a picture of someone texting and driving because that's thought of as a really relevant issue with multitasking, and it kind of shows how you're not able to fully attend to two things at once. So the resource model of attention basically states that we have a limited pool of resources to draw from when performing tasks, and that the amount of resources required to perform certain tasks determines whether or not they can be performed simultaneously. So something like driving obviously takes a lot of attention. You're looking, you're moving, you're steering, you're paying attention. There's a lot of things that go into being able to drive. You're listening for other cars, things like that. And texting, you're reading, you're typing, you're thinking of what to respond. So that also takes a good amount of attention. So that is why these two tasks are not something that can or should be performed simultaneously. So it's determined by task difficulty. So the more difficult either one or both tasks are, the less likely you will be able to multitask. Task similarity. So if two tasks are more similar, it actually makes it more difficult to perform them at the same time. So an example of this is like if you're writing an essay while someone is speaking to you, those are both like verbal input and output. So it's hard for you to write down something that you're thinking while someone is saying something different to you. So like if you like listening to music too when you write or read or something, at least me personally, I can only listen to instrumental music because I can't take in the verbal input from listening while I'm reading, which would also be like verbal input. I know that's a pretty common example of how task similarity can play into multitasking. And then task practice is kind of similar to task difficulty. So if you have a lot of practice with something and it's really easy for you and routine, that's not going to take up a lot of those resources, and that'll make it a little bit easier to multitask. All right, so question 9, this is going to be kind of review. So just testing knowledge on some of the topics that we've talked about earlier. So I'm not going to really go into these topics in any more depth after these questions, but recall is a great way to study, a great way to remember things. So this is just giving you a chance to do some recall from the information from today's lecture. So what kind of receptors do not encode for the duration of a stimulus? So take a minute here and pause the video. All right, assuming that you've answered the question, I'll show us the answer, which is C, those phasic receptors. So those are the receptors that only will fire at the beginning of a stimulus. And if the stimulus stays constant, they will not continue to fire, so they do not encode for duration. Tonic receptors would be the answer if the question asked what kind of receptors do encode for the duration. Photoreceptors and chemoreceptors aren't specifically one or the other, so those would not be correct. All right, so we have one more review style question for the end of today's lecture. And that question is going to be, the image to the right depicts which Gestalt law of grouping? Let's take a minute here and answer the question. All right, so our answer here is going to be A, the law of closure. So basically that's the idea that you see this as a triangle and not as three little circles, and then like the same with the background one, you see that as a triangle. All right, so thank you guys so much for listening to today's lecture. Again, I apologize if there was any meowing in the background. But so coming up next week on Sunday the 13th, we'll have a Psych Soc lecture about emotions and stress released at 8:30 p.m. on YouTube. Monday the 14th, we will have a biochem lecture about operons, post-transcriptional control, and cancer, released at 8:30 p.m. on YouTube. So if you have any questions about today's lecture or any previous lectures, feel free to send a message in that GroupMe that I had linked at the beginning. You can message me directly or throw it into the group, and we would be happy to help you out there. So happy studying. Thank you for coming.